Hello everyone, I'm going to talk about nucleotides and nucleic acids. The bDNA, we have three different types of DNA structurally. The one that is predominantly formed in the cell and is predominantly found in the cell is bDNA. As I mentioned, it is right-handed and it contains 10 base pair per helical turn, and it is the most stable structure of the DNA. It is main DNA found in physiological conditions. But of course, apart from the bDNA, we can have some minor forms of the DNA. One is aDNA, another one is zDNA. aDNA, again, is a right-handed, similar to bDNA. It is shorter and thicker. It has a shallower minor groove, a deeper major groove. It contains 11 base pairs per helical turn, and it is favored in many solutions that are relatively devoid of water. When you have low amount of water, the DNA it tends to form this aDNA form. Sometimes it is also formed when proteins attach to the DNA, because attachment of a huge complex of proteins on top of the DNA molecule makes it devoid from water, devoid of waters, and so it tends to form aDNA. And similar structure also is found in double-stranded RNA. Of course, we're talking about DNA right now, but if double-stranded RNA is formed in the cell, it's somehow similar to aDNA. And we also have zDNA, and which is in contrast to bDNA, it is left-handed, it is more slender, elongated. The minor groove is narrow and deep, and the major groove is barely apparent. It contains 12 base pairs per helical turn, and it is found where pyrimidines alternates with purines. It means that we have purine, pyrimidine, purine, pyrimidines alternatively. And it seems, it is suggested that these types of structures has regulatory role in DNA. Here in this picture, you can see the structure of aDNA, bDNA, and zDNA. This is the form of the DNA that is the main form of the DNA, and then it is the aDNA, this is the section, and the zDNA, which I told you is slender, and the section of the structure is also shown here. Process that can happen on DNA is denaturation. By denaturation, we mean separation of the two strands of the DNA. I told you that the two strands of the DNA are attached together by the hydrogen bonds that are formed between the base pairs. These hydrogen bonds are weak, but they can be. There are numerous of hydrogen bonds, and they can be separated from each other by, for example, changing the pH, and most commonly by heating the solution containing DNA. So if you heat the solution containing the DNA, the two strands will separate from each other, and this is called denaturation. When the two strands of the DNA are separated, we say that the DNA is denatured. You see, this is a process of the DNA denaturation. First of all, those places that are weaker, it means they contain AT base pairs because they contain two hydrogen bonds. They are weaker compared to the GC base pairs that contain three hydrogen bonds, and so they are stronger. So first of all, those places that contains more AT base pairs are separated from each other, and they're denatured. And then if the procedure of the denaturation is continued, then the stronger parts are also denatured, separated, and the whole molecule of the DNA is denatured, and it is converted to the single-stranded form. We have a melting temperature for the DNA. The strands of a given molecule of DNA separate over a temperature range, and the midpoint is called the melting temperature, or Tm. And Tm is actually the melting temperature at which half of the DNA molecules are denatured. And Tm is influenced by base composition of the DNA and by the salt concentration. And which one do you think has a higher Tm? DNA rich in GC pairs or the AC pairs, AT pairs? Of course, the GC pairs have higher TM because GC pairs have three hydrogen bonds, are bound together by three hydrogen bonds, but AT base pairs are formed by two hydrogen bonds. So you have to spend more energy to separate GC pairs. And that's why they are the locations of the DNA that contains more GC pairs are stronger. The DNA is packaged in the cells. In prokaryotes that do not contain a nucleus, the DNA is circular at the circle, so it does not have any free end, and it is supercoiled in this form so that the DNA is packaged and takes less space. In the eukaryotic cells, that the DNA is longer and it has two free ends, it is not circular. The DNA is packaged in the form of nucleosomes by the aid of proteins. And let me show you here. These are the chromosomes. These are the most compact forms of the DNA, and they contain chromatin. And you can see here, this is DNA that has been turned around the protein. This is the core protein, and the DNA around this core protein is called nucleosome. Twice, the DNA is turned around this protein twice, and these proteins are called histones. We have they contain 8 subunits: 2H3, 2H4, 2H2A, 2H2B. So from H3, H4, H2A, H2B, these subunits, each of them, each one, from each one we have two subunits. So a complete set of 8 proteins, 8 subunits make up the histone core for us, and then the DNA turns around this core of histones twice to make the nucleosome. Again, here you can see the structure of histones: H2A, H2B, H3, and H4, and the DNA that has turned around this protein. We also have a separate subunit called H1 that is present between the nucleosomes. So this is the histone octamer. Octamer means it contains 8 subunits, and then the separate H1 subunits. DNA packaging in eukaryotic cells is very, very interesting. And this is the DNA. It's shown by the magnification of 100 million. And this is the buried DNA. It does not contain any protein, but proteins come and attach to the DNA. And you see here that the core structure of the histone is being formed in the middle of the DNA. And so this is the nucleosome, which is formed by turning around the histone molecules. And then these nucleosomes can also make a three-dimensional structure. Here it is shown by the magnification of 50 million times. And this fiber that is formed is called 30 nanometer chromatin fiber. So it is the second order packaging of the DNA. It also forms a helical structure, which is shown here in the magnification of 10,000. And you can see there's a huge structure, but very, very organized. And again, we have some extra sort of helix structure, which at last makes up these chromosomes, which are the most compact form of the DNA inside the eukaryotic cell. And this form is only found when the mitosis is happening, and when the metaphasic chromosomes are formed in the middle of the cell before the cytokinesis or in the anaphase phase happen. And you see DNAs are moving towards the different poles of the DNA of the cell, and then the cells are separated and new cells are formed. This three-dimensional structure of the DNA is very important, and for example, some chemotherapeutic drugs like cisplatin only kill cells by uh, by disrupting this three-dimensional structure, they bind to the DNA, distort its structure, make a kink in its structure, and that's why the DNA, when it loses its normal three-dimensional structure, the cell will find it defective, and then will find it defective, and the cells will go through cell death. And that is exactly what we want to happen for the cancer cells. So cisplatin kills cells by disrupting this three-dimensional structure, and that shows us that the three-dimensional structure of the DNA is very important. Here you can see a summary of the comparison between DNA and RNA. The DNA that I explained for you is double-stranded. It contains deoxyribose as the sugar, and the major bases that can be found in the structure of DNA are thymine, cytosine, adenine, and guanine. In contrast, RNA is single-stranded, and the ribose in its original form, it contains the hydroxyl group, and it also contains the same major bases except thymine. And instead of thymine in RNA, we have the uracil. And so RNA does not contain any thymine. Another difference between RNA and DNA is that RNA is unstable because this phosphodiester bond can easily be cleaved by the aid of this hydroxyl group that is not commonly present in the DNA structure but is found in the ribose of the RNA. And the presence of this hydroxyl group that attacks to the phosphate and leads to the cleavage of the bond between the phosphate and the next sugar makes the RNA an unstable molecule. So in contrast to DNA that is very, very stable, RNA is very unstable and easily degraded inside the cell. RNA is single-stranded, but it doesn't mean that it does not have any base pairs, because it can form secondary structures. It can make these loops that are called hairpin loops, and these hairpin loops are formed by the base pairing between the complementary bases in one strand. We have different types of RNA inside the cell. We have the tRNA, which is its name is taken from transfer RNA. We have mRNA, and we have rRNA. We also have some small RNAs, like small nuclear RNAs, which is abbreviated as snRNA, and they are involved in splicing of the RNA. And we also have microRNAs or miRNAs, and also LNC RNAs or long non-coding RNA. And these two RNA types are involved in the regulation of gene expression. This is the structure of transfer RNA, and you can see that this is a single strand, but it has made some complementary base pairs in different parts. So it has 3 hairpin loops in its structure that are numbered as loop number 1, 2, and 3. And this is this 3' side is the accepting end of the amino acid, and amino acids are joined to this end, and so they are used as for the protein biosynthesis. And these transfer RNA molecules act as adapter molecules, because they, at one side, they read the code on the mRNA, and at the other side, they bring the amino acids that are complementary to the code that is being read. This is another depiction of the structure of the tRNA. You can see that we have many different uncommon bases in the structure of the DNA, like pseudouridine, thymidine, the dihydrouracil, and other methylated bases. And the tRNA is one of the extraordinary ribonucleic acids that contain these minor bases, the great amount. This is the structure of rRNA. As you can see here, it contains a very huge structure, and we have many different types of rRNA molecule. Again, I just want you to have an imagination of the rRNA structure, and there is no need to memorize these structures. And then we have the messenger RNA. It is called messenger because it reads the message on DNA at the template. So DNA includes the informations at the genes, and mRNA is a transcript of these information, and then mRNA is transcribed from DNA, and then it is exported out of the nucleus into the cytoplasm, and there it is being used for making the protein based on the code that it contains. In the eukaryotic cells, the RNA that is first transcribed from DNA contains the information from both introns and exons. But then the parts that belong to the introns should be removed. So the first transcript of the RNA is called hnRNA, and then through the splicing mechanism, the introns are removed, and the places and only the sequences that belong to the exons will remain, and so the mRNA, the mature mRNA, will be formed, and this procedure is called splicing. The splicing means removing of the introns on the mRNA molecule. For mRNA processing in eukaryotes, we first add a cap to the 5' end of the mRNA. This is a cap which contains 7-methyl guanosine, and then this cap is bound to the mRNA by the extraordinary 5' to 5' phosphodiester bond, and then we add a poly-A tail. It means that by poly-A tail we mean a tail on the 3' end of the molecule that contains many adenine molecules. And then we remove the parts that belong to the intron, and the exon, the sequences of the exons will be remaining. So they attach together and make this mRNA molecule that is exported out from the nucleus into the cytoplasm. I hope that you have learned this part, and if you want to have further reading, you can take a look at the biochemistry books. Especially, I recommend Leninger textbook of biochemistry. Have a good day ahead.